Welcome to the presentation on plant breeding. So let me share the screen. And for plant breeding, there's one PowerPoint, and this is it. So why do I bother with plant breeding? This is not a breeding class. This is a vegetable crops class. Well, two reasons. One, breeding is my area of expertise, my passion, my career up till the point I came here to Hartnell to start teaching. And I really feel there's an important value in understanding a little bit about genetics and a little bit about what goes into these varieties. We talked about crop selection already, and you have to make a decision. Am I going to grow some crisphead or iceberg lettuce? Am I going to grow romaine? Am I going to grow broccoli, carrots? What are you going to grow? But even when you decide what you are going to grow, you have another major decision. Which variety of what you are going to grow? Which genetic makeup of what you are planning to grow? And that's really important. And it is a compromise no matter how you cut it. You've got to compromise between the performance of the potential quality. If everything goes perfectly, what is the potential quality for that variety? What can it do? What can't it do? What are the risks with diseases and pests? Uh, some pest or disease issues, there is no variety that really helps you. You can have maybe a more vigorous uh, crop that's somewhat less susceptible, but overall, there's no true resistance to the uh, the pest or disease in question. In other cases, there are choices. Is the pest or disease that you are likely to have, or at least at risk for having in that crop, in that location, is there something available that has a resistance to it and has the other qualities that you're looking for? Now, I mentioned the word resistance. Don't forget, resistance is a yes or no answer. It's either resistant or it is not. It either can get the disease or it cannot get the disease. At least cannot get the disease under normal real world conditions. Sometimes you can overcome a resistance in the lab or something. But true resistance, yes or no. Tolerance, that's a different word. Tolerance is about a level of a plant's ability to survive and reject or inhibit the pest or disease long enough to minimize or prevent damage until you can harvest it. And it's important to be truly tolerant. It has to allow you to get a quality, marketable crop harvested and to market. Just because something is only 50% melted down when the neighboring crop is, you know, 70% melted down or 90% melted down from some horrible fungus, that's not tolerance if you don't have anything left to sell. Anyway, just some important vocabulary. So, what is breeding? Breeding includes controlled pollinations where you intentionally choose plants to behave as the mother and the father. The mother is going to receive the pollen from the father. And so you pick the mother and father plants such that they will hopefully give children that are genetically superior to either the mother or the father. And so it takes a lot of knowledge, a lot of time, a lot of patience to do those things. Then you have to do selections. So a plant breeder might look at hundreds or even thousands of crosses, and they have to grow them out and evaluate them. Maybe some early evaluations might be just a few plants, and maybe just one plant. And maybe they are going to test them by spraying mold spores on them. And if the mold spores infect and the plant dies, it doesn't go on to the next generation. If it survives, then it goes on to the next generation. That's just one type of selection. But you're looking at all sorts of things, color and size and leaf shape and root structure and whatever's important to the breeder for that situation. Testing is when you are doing those uh, evaluations, either spraying a mold on it. Maybe I'm taking samples. If I'm breeding tomatoes, I'm taking samples of the ripened tomatoes after I've grown them out. And I'm measuring pH and I'm measuring the weight, I'm measuring the diameter, I'm measuring the color, I'm measuring the soluble solids content of the tomato juice. I'm testing the viscosity or thickness of the pulp from the tomatoes, and on and on and on. Again, what are you testing? What are you looking at? What are you measuring? It depends. What are you looking for? What is your customer looking for? So controlled pollinations, there's wind pollinated crops like corn. So you have to go through, and these male tassels, which contain the male flowers that are separate from the female flowers, and they grow up and form these tassels above the corn plant and sprinkle pollen throughout the region on the wind. And so you need to take the females and detassel them. You don't want the females making pollen. So before they're mature, you rip off the tassels, detasseling them. And so the females are only going to have the female flowers with the silks coming out. Spinach, that's really tricky. I did spinach breeding and spent many thousands of dollars doing construction of special aluminum frames that were covered with custom sized and made plastic bags that were, I don't know, maybe about the size of a 55-gallon drum. And we would have two plants each in every one of these chambers, maybe 100 chambers that I had at a time, and special ventilation system going to it with a HEPA filter, a very high grade hospital filter, but huge, like hospital isolation room kind of filter. So no pollen can get out of those chambers and into another one of the chambers. So when I put a male plant and a female plant into that bag with the aluminum frame, covered with the bag, it's only going to be those two mating. A lot of work, a lot of tedious work. Lettuce, very different, but it's not wind pollinated, so you don't have to worry about it. So those are two wind pollinated crops. Insect pollinated crops include the brassicas, the cabbage, broccoli, cauliflower, etc. And a lot of the brassicas have what are called self-incompatibility. In other words, even though they're being insect pollinated, if they have the self-incompatibility gene, they need a different SI gene in order for the pollen to work. If it has the same SI gene, it won't work, even if it came from a completely different plant. It's these SI genes that recognize any pollen that comes from itself or something that is too genetically similar, at least with the SI gene. And that promotes outcrossing. So you need different SI genes, self-incompatibility genes, in order to cross. And so breeders need to think about that. How can that be used to their advantage? And what are the disadvantages that they must overcome in order to breed and make the crosses they want to cross in that brassica? A lot of crops, you need rented bees, and that's an issue that has to be thought out, planned. You've got to contract with a a beekeeper and make sure you're well loved by your beekeeper, because if they have a disease or other problem and they get short on bees, who's going to get the bees that are left? Self-pollinated crops, tomato is self-pollinated, but you can do crosses that require emasculation. Use tiny little tweezers and you pull off the staminal cone that has the stamen and the um, pollen inside. And before they've matured and burst to release the pollen, you go through with a crew and remove the pollen from all the females. And then all the males, you go through and take a vibrating tool. I just used a modified electric toothbrush and put on the end of the electric toothbrush a little spring, a little larger than the spring inside of a ballpoint pen, and slipped inside of that spring half of a capsule, the same kind of capsule that you would use to make medicine, those little plastic capsules that are filled with some sort of medication, whatever it is. You can buy the capsules empty. You take half of the capsule, stick it into the inside of the spring on the flowers that you have not emasculated, that you want to be males, and the pollen will be shaken off the anthers into the little capsule. You can put the other half on the capsule, put it in a labeled test tube, and send that to storage and refrigeration till you need it later to pollinate your flowers. Lettuce, there is no way to emasculate. It's too complex. It's just too challenging. And so that's a really serious limitation. And so whereas tomato has huge labor to make hybrid seed, lettuce, you can't make hybrid seed because there's really no practical way to do it on a large scale. And all of the pollinations have to be done by hand, and they have to be done by hand in a very narrow window early in the morning. And I had some crossing days, I'd be there at 6 in the morning getting things set up, getting all my flowers ready, tagged, and then all of a sudden things start happening. And the flowers that were opening quickly, I would have to put them in a cold area to hold them so they don't um, flower too quickly and their pollen get ready. And the flowers that were a little slow to develop, I put them in the brightest, warmest part of the greenhouse. I might even use the refrigerator for some to hold them till the other plant was ready. And a lot of work, a lot of effort. And then once the pollinations start, it could be 30 minutes of actual boom, 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 boom work, and you're just going crazy. And then it's too late. Your 30 minutes, maybe an hour is up, and you're done pollinating for the day. You can set up, set up was a couple of hours of work, but the actual pollinations, 30 minutes to an hour, done for the day. You can't do anymore. Just the way lettuce works. And you got to know your crop. How am I going to get pollen from one flower with characteristics that I like, that I want to be my male, onto another flower with other characteristics that I like, that I chose to be the female? Lots and lots of challenges. There are also clonal crops. You're not constantly making new seed. Once you have the perfect plant, the one that's performing the way you want, it has the beautiful strawberries or the beautiful potatoes or whatever you're making, and you found that perfect plant, then you keep reproducing it through clonal propagation. Potatoes, you grow a plant of potato and you get a bunch of potatoes. You save a few of them, and those are called seed potatoes, the ones you saved for seed. And you can save the defects. They're genetically not defective. They just are defective because maybe they hit a rock in the ground or whatever. All of the potatoes don't develop exactly the same. And If you grow one from the same plant, it'll give you a genetically identical plant, unless there was some bizarre, highly unlikely mutation in the plant itself, and that's really not very likely. Uh, so now you can just keep growing it. Once you've scaled up, you can scale up very quickly with seed potatoes over a few generations and then have enough to sell. The same type of thing is true for strawberries. Strawberries are genetically very complex, and you really can't reproduce a new strawberry without, or excuse me, let me say that again. I misspoke. You can't reproduce a new strawberry that you've developed twice. I can't make the same cross twice. You're not going to get the same thing. So you're finding one individual. That's your perfect individual, your mother plant. And the strawberry breeders that I used to know, I wasn't doing strawberry breeding for too long, so I never had this problem. But some of them that had their cash cow mother plants, they would keep them in high security, um, vicious guard dogs, and have a shotgun on site. It's serious business, because that one plant might literally be worth $500,000. One plant, because it is the genetic mother to all of that variety being sold. And so you take runners from that one plant, and the runners from that one plant are clones. They're just little plantlets that grow out on these runner or lateral stems. And then you plant them in an area that promotes runner formation, which in California, that would be the area near Mount Shasta, up in the north, Susanville, north and east of Redding, where you're going up in the hills, snow country, not too high, but certainly where it gets cold. And strawberry runners produce really well in that environment. So again, you need to know your product. You need to know that I can do breeding lots of places, but if I'm doing strawberries, I got to make my runners way the hell up in uh, the mountains north and northeast of Redding, near Mount Shasta. And then I need to know that all of those processes for harvesting those runners, packaging them, and getting them to my customer, it's challenging. And I've got pollinations for breeding. I described some with lettuce, where I'm busy trying to get the things matched up exactly right to do the cross from mother to, or excuse me, from female and male to cross the pollen. And for growing seed to sell to the farmer, lettuce is super easy. Once I have the plant that I want, I just plant it out in my seed field. I plant a bunch of seed from that plant and grow it up, let it do itself. It's self-pollinating, and it will do itself without me having to worry about anything. Pollinations for breeding in tomato, lots of work as well. But the hybrid seed in tomato, also lots of work. So that's kind of the disadvantage. Lots of work, very expensive. So production seed and breeding research seed, very different things you try to do. And with that, I am going to take a short break.